So far, what we've learnt about the various factors that might influence the stability of this double helical folded structure of DNA has been less than encouraging. So first we thought about the entropy, which is this universal tendency of energy to become more widely dispersed into different forms. And we saw that in chemical structures this tends to correlate with favouring disordered structures, where a large number of different arrangements of the atoms can be sampled, because if you sample a large number of different atomic arrangements, then you have lots of different ways of distributing the energy. So an unfolded state, where the chain has lots of freedom to sample different shapes, would seem to be more favourable than this rather well-defined folded structure. Secondly, the backbone, which, remember, is dominated by these phosphate groups all the way around on both of the intertwined strands, that too is going to struggle to achieve this conformation, because winding it up into this kind of structure brings the negatively charged phosphate groups closer together than they would tend to be in an unfolded state. And the repulsion between them, that electrostatic effect, again, is going to favour unfolding. So then we looked at the hydrogen bonding involving the bases in the middle of the structure. And we saw that that probably is really just about breaking even. That in the unfolded state, those bases can hydrogen bond to water. In the folded state, some of them are taken away from the water, and it's then necessary for them to hydrogen bond to each other. Otherwise, for sure, the double helical state would be destabilized. But if the bases do succeed in making complementary hydrogen bonds, to a first approximation, all those do is replace hydrogen bonds to water. So again, they don't seem to be driving the folding. So really, we're left with an increasingly desperate drive to find something which can account for the stability of this structure. And it has to be with the one thing we haven't really thought about yet, which is the stacking of the base pairs. So this one parallel to this one, parallel to this one, and so on, all the way up along the axis of the structure. Now remember what we said before, which is that this familiar representation of the structure, where we basically just show the bonds and we don't attempt in any meaningful way to reflect the volume occupied by the atoms, that this representation is misleading, because it makes it look as if there are these vast empty spaces in the middle of a very sort of open structure. But if we change that representation just for the bases in the middle there and attempt to give a meaningful volume to each atom, we see something very, very different, which is that that core structure is basically filled up all the available space. In other words, the helix has twisted up just enough so that those bases are stacked flat on top of one another with essentially no space in between. So those interactions, the stacking interactions of the bases, are what we need to now examine and see whether we can interpret the stability of the structure in terms of those. Let's take another look then at what these bases actually look like. So this is adenine. So remember, we have this aromatic system and it's therefore flat. If we turn it around like that, you can see it has this dead flat structure. And all round the fringes we have these polar groups with the blue nitrogen atoms having very different electronegativity from the carbons and hydrogens that they're attached to. And we can see the consequences of that quite clearly if we generate an electrostatic potential surface. So if we do that, if we look at the edge of the ring, you can see it looks extremely polar. So we've got delta negative, where we have a nitrogen in the ring, and then delta positive, where we have NHs. And you can see all the way around, it looks really quite polar, as we would expect. But if we look at the flat face of the ring, you can see that while you can still see the polarity around the fringes, there are big areas in the middle which are actually not polar or hardly polar. So there's a real difference in character between the essentially non-polar flat faces and these fringe groups with all the nitrogens and NHs and so on attached. And we can see the same thing if we look at any of the bases. So here is guanine, where again you have this 10 pi electron aromatic system, and again if we generate the electrostatic potential surface, you can see on the flat face lots of non-polar surface across here, but round the fringes very polar, and therefore if we look at it edge on, it looks very different and it looks like a very polar structure. Okay, so taking that characterization into account, let's start to look at what the possibilities are for the forces that could exist between this pair of bases and this pair of bases when they're stacked on top of each other. So it's the forces acting this way that we're interested in. Well, one thing that we can say fairly confidently is that there won't be any hydrogen bonds, because the thing about hydrogen bonds is that they have quite strong directional preference. So here we consider two hydrogen bond geometries. So we've got, as an example, an NH covalent bond and then a hydrogen bond to a delta negative oxygen. And same here, but now we've put the hydrogen bond at a right angle to the covalent bond. And it turns out that this geometry is strongly preferred to this one. And the reason for that is fairly obvious when you think of it just purely in terms of electrostatics, because a hydrogen bond is primarily an electrostatic bond. It's not quite as simple as that. There's some sort of electron sharing involved so that it kind of hovers on the fringes of being a little bit covalent, but it's primarily an electrostatic interaction. And you can see that if you're this delta negative oxygen, this is the best position for you, because this way you get as close as possible to the delta positive hydrogen, which you're attracted to, but you've also got to take account of the fact that you want to stay as far away as possible from the delta negative nitrogen, which you're repelled from. And this is the geometry which does that best. If you do this, the distance from the delta positive hydrogen is the same as it was here, but this time you're much closer to the other delta negative nitrogen. So the repulsive term across here is going to be much stronger than it would be in this geometry. So for that reason, hydrogen bonds favour this linear geometry. Now it's not absolute in the way that it is for covalent bonds. You're used to the idea that for covalent bonds, you know, you can work out the bond angles from electron pair repulsion theory or whatever, and that will be what they will be in the molecules because it's such a strong preference. Here, because the bonds are weaker, you don't pay such a big price for deviation from the ideal. So in reality, if you look at the angles formed by hydrogen bonds in crystal structures of biological molecules and indeed other molecules, this is certainly the preferred angle, but some variation around here is observed. Now, the other thing that you have learnt about hydrogen bonds is that they tend to be directed not just generally at the delta negative atom, but specifically at lone pairs. So here's an example of two water molecules hydrogen bonded together, and what you have already learned is that where you have the OH covalent bond here, that hydrogen tends to be hydrogen bonded specifically to a lone pair on the oxygen here, which is what this represents. And again, that makes sense in terms of electrostatics, because this is where the electron density is focused, and therefore where the negative charge that the delta positive hydrogen is being attracted to is focused. So it makes sense that hydrogen bonds should point at lone pairs. This, it turns out, is a much more controversial area, because what you find in structures is that sometimes they do, but sometimes they don't. So in protein structures, for example, where one of the common ways that proteins fold up involves forming helical structures called alpha helices. If you look at in those structures, the hydrogen bonds which play a role in forming those helices do not point at the lone pairs of the oxygens. They kind of point right in the middle, in between them. So this seems to be a much less absolute requirement. But again, it's a preference. So if we come back to think about how this is going to work with the bases in DNA, remember their defining characteristic is that they're aromatic and therefore planar. And that has a huge knock-on effect on the hydrogen bonding, because this structure is planar, and also this lone pair on the nitrogen here, you've learnt about this in organic chemistry, that where you have a nitrogen or an oxygen directly bonded to an aromatic ring, the lone pair becomes delocalized into the aromatic system, and that's what's going to happen here. And of course that requires this lone pair to be in a p orbital which is parallel with the p orbitals that form the aromatic structure here. And that in turn means that the geometry around this nitrogen is going to be effectively the same as it would be if this were a fully fledged double bond, because in order to keep that lone pair aligned, it has to in effect behave as if it were forming a full double bond there rather than just a partial one, and therefore the geometry is going to be the same as it would be around a doubly bonded carbon, which means of course that it's all planar. It's a trigonal planar nitrogen around there. So these hydrogens are also in the same plane as the aromatic ring, and of course these hydrogens too coming off the ring are all going to be in the same plane for the same reason, that in this case you've got trigonal planar carbons, because they have to be trigonal planar in order for their p orbitals to participate in the pi bond. And in the case where you have NHs coming off the ring, as here for example in guanine, again that nitrogen, remember, although it doesn't look it the way we've drawn out the structure here, if we think about the resonance structure where this is a double bond, then this nitrogen also has to be trigonal planar, and therefore the hydrogen will all be in the same plane. So that's the simple picture. All the atoms are in the same plane. Okay, so here is a slice through the actual crystal structure, showing in this case a GC base pair, and we can see the three hydrogen bonds across the middle there. So we've got NH to carbonyl here, and you can see it is collinear. So you've got nitrogen to hydrogen covalent bond, and the hydrogen bond does point collinearly with that. Here we've got NH to delta negative nitrogen, and again it's collinear with the NH, and the same here with the NH to carbonyl. We can also see that the hydrogen bonds are pointing in the directions we would expect in terms of the lone pairs, because here we've got NH pointing at a carbonyl at that sort of angle, and remember, two lone pairs on the oxygen we would expect to be one to be pointing that way, one to be pointing that way. So it is pointing directly at one of the carbonyl lone pairs. In this case we've got NH to nitrogen. Nitrogen has only one lone pair, and it's going to be pointing directly out this way. So again, the hydrogen bond is pointing directly at it. So the Watson-Crick base pairs do a very good job of meeting the hydrogen bonding requirements. And all of the hydrogen bonding potential in terms of the NHs and the delta negative nitrogens and oxygens with their lone pairs, all of that potential is contained within that plane. So between this plane and the next one, there will, as we said, be no hydrogen bonding to worry about. There is something else quite striking about the base pairs, which we can see quite clearly if we look at them from a different angle. So let's just hide the backbone so that we can just focus on the base pairs. So there we are. And if we look at this pair, for example, so there you are, you can see the two bases there with the hydrogen bonds in this region here. But if we look at them along the axis of the base pair, you can see quite clearly that actually the whole thing is not flat, that one ring has got a slight twist on it relative to the other one. And if we look at all the base pairs, you can see that that's always true all the way down. So here again, you can see that twist between the ring at the front, which is angled like that, and then the ring at the back, which is vertical. And it's the same all the way down, so you can see it there and there. And that's called a propeller twist, because it's the same as if you look at the two blades of a propeller, that they're angled relative to one another like that. The consequence of that propeller twist is that the Watson-Crick hydrogen bonds are actually not quite as good as they might be. So that distortion makes them not quite as linear and short as they could be, and therefore their energy is raised. So there must be some factor which more than compensates for that and makes it worthwhile slightly distorting the hydrogen bonds. So again, there must be a clue here as to what is making the helix stable. If we take the structure and rotate it round so that we're looking end on down the axis of the helix, it becomes very obvious that the base pairs are not stacked directly one above the other. So let's look at that in a bit more detail. So here is a single base pair at the top of the helix, and let's change this to a space-filling representation first of all. So there we have one base in blue, the other one in red. You can see the line where the hydrogen bonds are down the middle. And let's then just move this slice down through the structure. So if we carry on like this, you can begin to see the next base pair appearing underneath. And the important thing is that, again, you can see that they're not directly aligned one above the other, so that instead of this one being entirely hidden underneath, you can see a corner of it on each side, which is not hidden by the base pair on top of it. And if we keep on doing that, we can begin to fade out the top one, and now we can see this base pair and then the one underneath it. And again, they're not directly on top of one another, so always you get a corner which is left exposed as a result of this orientation. And here, for example, the methyl group from a thymine base, you can see clearly is left exposed in the major groove because the base pair on top of it is not completely aligned. Now, if we ask why the bases should stack in this sort of offset way, we have to remember that they're not free agents. They're connected by the backbone. And if we tried to rotate them so that they were more perfectly aligned, so that there was greater contact surface area between the base pairs, then it's going to affect the backbone. And you can see that in the structure as it actually is, the backbone is really quite extended. In other words, it's stretched out, close to as much as it could be. And that's important because, of course, it keeps those negatively charged phosphate groups apart from one another as far as they can be, and that reduces the energy. So if we tried to rotate a base pair around, so for example, if you focus on these two, we would have to rotate this base pair around that way to bring it into alignment with this one, and then we'd have to bring this one around that way a lot more to bring it into alignment as well. And you can see what that would do is distort the backbone structure, or else if we simply unravel the helix, the bases would no longer be in contact at all. But if we forced them to stay in contact, it would distort the backbone structure. And that might be possible, but it would certainly bring the phosphate groups closer together and therefore be unfavorable. So the offset is a result of the bases being connected by the DNA backbone there. Now, if we come back to the issue of the propeller twist, if we represent it schematically like this, where each of these rectangular blocks represents a base pair, on average, the angle between one base pair and the next is around 36 degrees. It varies a bit within the structure, but it's in that region. And we can see the consequence of that is these areas here and here are the ones where they're not packing against one another, where those remain exposed to the solvent water. And we can gain some insight from this into the propeller twist. So the diagram at the bottom, you have to imagine you're looking along this line, so along the line that runs through the middle of one of these bases. And this is what it would look like if there were no propeller twist. So you can see that there's really quite limited overlap between the bases at the corners there. But if we rotate them round slightly, so that the lines running through the middle of them are kept in the same place, but we simply rotate each one around a little bit like that, then you can see that the effect of that is to increase the contact area between them. And it seems likely that that's the reason for the propeller twist, that we pay the price of weakening the Watson-Crick hydrogen bonds a little bit because we gain better surface contact between the bases with that rotation. So that's further evidence that the stacking of the bases on top of one another, that surface contact between them, is of huge importance in stabilizing the structure. So that brings us at last around to the key question of what it is that's driving that stacking process, that's making it favourable for these flat surfaces to pile up on top of one another. So this was our electrostatic potential energy surface, looking down here on an AT base pair. And here is the hydrogen bonded interface between them. And what we emphasized before was that although there are lots of polar atoms, they're all focused around the outside of the individual bases, but that a lot of the surface on top has really very low electrostatic potential. And that is important because what we're therefore talking about to a large extent is the packing of more or less non-polar surfaces on top of one another. And that should bring to mind the idea of induced dipole-dipole forces, or London forces, or dispersion forces, whatever you want to call them, which are those universal forces that can act as attractions between any kinds of atoms. And one possibility, therefore, would be that it's those kind of forces operating between the base pairs which are driving the structure to fold up. And one thing that's very important here is the distance dependence of that attractive interaction. So what you can see in this graph is a comparison of the distance dependence for a classic electrostatic attraction, in other words, between point charges, and that's what this brown line represents. So if you have, for example, a fixed positive charge and a fixed negative charge, the potential energy decreases as they get closer together, obviously because of the attractive force, but it's quite a gradual thing. So you can see that it varies as 1 over the distance between them, which gives you this sort of rather gradual decrease in potential energy as they get closer. And that's important because it means even when they're not directly in contact, they can still be making a significant contribution to a favourable potential energy. Equally, of course, when you have two like charges like the phosphate groups, even though they're not directly in contact, the repulsion between them, as we've seen, is significantly destabilizing the double helical structure. However, when we come to the induced dipole-dipole force, which is represented by the blue line here, the big difference is that this depends not on 1 over the distance, but on 1 over the 6th power of the distance. And that means that it only operates at extremely short range. So by the time you come up to this point, it's virtually negligible. And that's very important because what it means is that those surfaces pretty much have to be in contact. Otherwise, the induced dipole-dipole force will be negligible. And that's one possible factor dictating the double helix, forcing the two helices to screw down so that the bases are stacked directly on top of one another in order to maximize that induced dipole-dipole interaction between them. Of course, they can't get too close because then you get a very strong repulsive interaction. The repulsive interaction between atoms, you can see this in the bottom curve here. This is called the Leonard-Jones potential curve, and it's just a sort of empirical curve which builds in the r to the minus 6 dependence down here of the attractive force, but then the fact that when the atoms get very close, you get a repulsive force between them. And it's not that they're completely hard spheres, so it doesn't sort of just hit a brick wall here, but you can see it is extremely steep, and it's modelled typically as being dependent on 1 over the 12th power of the distance between them, so it's an extremely steep repulsive term. And that results essentially from the fact that if you bring the atoms too close, you're effectively trying to cram electron pairs into the same orbitals because you're overlapping orbitals there. And that causes a huge increase in potential energy. So there is this very well-defined, quite narrow range down here at the bottom of the curve in which it's favourable to maximise the forces between atoms. So we should think that that may be a significant factor here. But it is more complicated than that in DNA because, of course, we're not just talking about a pair of neutral atoms. First of all, we know that the base pairs are aromatic. And that's important because it means that when we bring those flat surfaces in contact with one another, what we're actually bringing into contact primarily are the delocalized pi electron systems. And so what we really should be thinking about a bit more specifically than that is the interaction between pi electron systems. And secondly, of course, we know that it's not completely nonpolar, that there are differences in electronegativities between the nitrogens and oxygens and the carbons, and that puts some partial charges in place there across that face. So even though they can't form hydrogen bonds between the stacked up base pairs, they can nevertheless give a sort of permanent electrostatic contribution. This issue is made very clear by studies of a much simpler system, which is just benzene. And it's been found that in the gas phase, benzene can form dimers, where two molecules stick together just through non-covalent interactions. And simple ideas based on induced dipole-dipole forces would lead you to think that this would give you the greatest contact surface area between them, and therefore would give you the most favourable geometry for the dimer. But if you carry out quantum mechanical calculations, what you actually find is that if you slide this ring up this way, to create a structure that looks more like that. This is what happens to the potential energy, which is that actually this displacement of one ring reduces the potential energy to a minimum with a displacement of about 1.5 angstroms. 1.5 angstroms corresponds to this kind of displacement of one ring relative to the other, and then it starts to increase again. So that suggests that there's something else going on here, that the interaction between them is not a simple induced dipole-dipole interaction. In fact, the experimental studies suggest that neither of these structures is what's predominantly formed in the gas phase, and what you actually get is something that looks more like this, a sort of T-shaped configuration. And you can see here that actually that's really looks very unpromising from the point of view of contact surface area, because it's really only a point of this that's in contact with the ring below. So the question is, well, what's going on here to make these surprising results happen? We can gain some insight into this if we calculate the electrostatic potential energy surface just for a simple benzene ring, which looks like this. So here, red corresponds to a negative potential, and blue corresponds to a positive one. So what's happening here is really two things. First of all, carbon is somewhat more electronegative than hydrogen, although it's not a big difference. There is some difference, and that's why these hydrogens around the outside are all delta positive. That's really just an electronegativity effect. But the delocalization of the pi electrons gives you a cloud of pi electrons above and below the ring surface, and because of the circular shape of that pi electron cloud, the electrostatic potential is focused sort of in the middle of it. So the most negative place, if you like, is in the center of that pi electron cloud above and below the surface of the ring. So if the two rings packed face to face, which is what just thinking about induced dipole forces would lead you to think, you can see that the problem there is that these two regions of negative potential are directly in contact with one another, and that's going to give you a significant repulsive force, and that's what would tend to push the ring off to the side, because you can see with the displaced arrangement like this that now the center of negative potential here is aligned with a positive hydrogen at that point, and that gives you a more favourable electrostatic energy. The T-shaped one again does the same thing, because again you get the delta positive hydrogen placed directly above the centre of negative potential. So in the context of DNA, this just makes the point that we shouldn't just be thinking about what maximizes the contact surface area between the base pairs, but potentially what maximizes the electrostatic contribution to the stacking energies. There have been a lot of attempts to use quantum chemistry to investigate this stacking of base pairs on top of one another. So here's an example. So what they did here was to calculate the energy of interaction between particular combinations of base pairs. So here, for example, you've got an AT pair stacking on top of another AT pair, and they've calculated the energy of that interaction as a function of the angle between the two bases. So here, 0 represents the bases directly on top of one another, as they would be if we unraveled the DNA helix a little bit, as we discussed earlier. And then the first point after that, which is here, is at 36 degrees, so that's close to the actual angle seen in DNA. After that, these angles would not be accessible to a DNA molecule unless it radically changed its folding, because you can't achieve that with a double helix because it would be too twisted. But I guess with other kinds of structures, in principle, you could get these other angles. But really, we're mainly interested in comparing the 0 and 36 degree points. And you can actually see for every one of these combinations of base pairs, 36 degrees gives you a lower energy than 0. So that's important because that says that it's not just induced dipole-dipole forces, because they would be greater at 0 degrees where there was complete overlap. And so that supports the idea that the electrostatic effect is making an important contribution to the stacking energy of the base pairs. The other thing, though, that's worth noting is just how different these pictures are for the different combinations of base pairs. And that's something that we've kind of ignored before, in that we've just talked about a base pair and another base pair as if they were all the same. And of course, they're not. And once you bring in these electrostatic effects and polarizability effects, the ability of one base pair to affect the electron distribution in the other one, that's also important. Once you bring those in, then of course it makes a big difference which particular bases you've got. And even if we just focus on those first 2 points, which is the difference between directly above one another, so 0 degrees, and the 36 degrees that you get in the DNA structure, you can see that if you have an AT above an AT, it makes an enormous difference. Whereas if you have an AT above a TA, it makes a much smaller difference. So these stacking effects are sequence dependent, and that makes the whole picture a lot more complicated than it would be if the DNA structure was just completely uniform. So there we are then. We have identified the stacking interactions between base pairs as something that could potentially be very important in holding the double helix structure together. What we haven't done, though, is what we said we must do when we were talking about hydrogen bonds, and that is, first of all, to think not just about the folded state, but about the unfolded state, to compare those intermolecular forces we've identified here with what the bases would be doing in the unfolded state. And in order to do that, what we also have to think about, which we haven't done here, is the solvent water. So where we'll go in the next section is to put that right and to bring in a consideration of what happens to the water surrounding the DNA when the DNA folds up, and we'll see that that makes a really important contribution to stabilizing this structure.